Bismillahirrahmanirrahim. Today we will discuss the process of aerobic respiration. Aerobic respiration consists of three main steps: glycolysis, Krebs cycle, and electron transport chain. In a previous lecture, we have discussed the process of anaerobic respiration, in which we have discussed the details of glycolysis. As we know that in glycolysis, glucose is broken down into 3-carbon compound, that is a pyruvic acid. So this in aerobic respiration as well as in anaerobic respiration, glycolysis share both the processes. So, but the difference is that, which we have discussed in previous lectures, that pyruvic acid, in the absence of oxygen in anaerobic respiration, move into the fermentation metabolism and produce lactic acid or alcohol. A small amount of ATP is produced in anaerobic respiration, while in the aerobic respiration, pyruvic acid will enter into the mitochondria. So, in aerobic respiration, pyruvic acid will move into the mitochondrial matrix, where Krebs cycle will be operated, and in the next slide we will discuss how that enter into the mitochondrial matrix. So here we will discuss the entrance of this pyruvic acid into mitochondria, that is known as oxidative decarboxylation of pyruvate, in which that pyruvic acid, which is a 3-carbon compound, it will enter into the mitochondrial matrix with the help of this carrier protein, which is a transporter protein, which is present on the mitochondrial membrane, through which that pyruvic acid will enter into the mitochondrial matrix, where there is a mechanism in which carbonyl group is removed as a carbon dioxide. Here carbon dioxide will remove here. Then pyruvate is oxidized to acetate. First it will convert it into acetate, where this high energy compounds in the form of NADH will be produced. Here NADH in its reduced form. So NAD will be converted into NAD. That's why it's known as oxidative carboxylation, in which carbon dioxide is removed and one NADH is produced. And in the last step, coenzyme is added into the acetate and acetyl CoA is produced here. So one molecule of the pyruvic acid, when enter into the mitochondrial matrix, it is converted into acetyl CoA at first before entering into the Krebs cycle. So it is a very important step, and this whole process is carried out by the enzymes pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex consists of 3 enzymes named as E1, E2 and E3. The details of this will be discussed in separate lecture. So as the pyruvic acid enter into the mitochondrial matrix. It is converted into acetyl CoA before entering into the Krebs cycle. Here, one carbon dioxide is produced and one NAD is reduced in the form of NADH. So, let's discuss how that acetyl CoA will enter into the Krebs cycle. So here we will discuss how that acetyl CoA enter into the Krebs cycle. Before starting the Krebs cycle, we will discuss a different steps. Krebs cycle is consist of 8 different metabolic steps, and these steps are divided into 3 phases. First phase is a preparatory phase, second is the decarboxylation phase, and last one is the regeneration of the acceptor of acetyl CoA. So let's start with the entry of acetyl CoA. Acetyl CoA, in the first reaction, acetyl CoA will react with the oxaloacetate or oxaloacetic acid. It is a 4 carbon compound, and this acetyl CoA are a 2 carbon compound, and with this reaction in the presence of citrate synthase enzyme, it will convert it into citrate. This is a 2-step reaction in which at first oxaloacetate react with the acetyl CoA and complex known as citryl CoA is formed. Here citryl CoA, and then by adding water, this citryl CoA will be broken down into coenzyme and citrate. So here citrate is formed. Then this citrate will enter into its preparatory phase, because here the process of decarboxylation will be started, but this citrate cannot start the decarboxylation process. It must be rearranged into its isomer, which is a isocitrate. That is a rearrangement here. This citrate will be converted into isocitrate with the enzyme, a conitase enzyme. Here it is also 2-step reaction in which citrate will be converted into isocitrate, so that may easily be decarboxylated. Here isocitrate, in the presence of enzyme isocitrate dehydrogenase, will be converted into alpha ketoglutarate, and let's see, one of its here carboxylic group is removed in the form of carbon dioxide, and in the same way hydrogen removed is captured by NAD, and NADH plus H is formed here, and isocitrate is converted into alpha ketoglutarate. Here again alpha ketoglutarate, in the presence of enzyme alpha ketoglutarate dehydrogenase. It's a first dehydrogenase, and this is a second dehydrogenase enzyme. The same way it also removed carbon dioxide. One of its carboxylic group is removed in the form of carbon dioxide, and hydrogen removed, and one NADH is produced. NADH plus H is again produced here. So here one of in place of that carboxylic group, the coenzyme which was released here was enter into the reaction, and succinyl CoA is formed at this step. Again this coenzyme, which has been removed from the acetyl CoA or citryl CoA, will enter into the in this step, and succinyl CoA is formed. And after that, succinyl CoA, in the presence of enzyme succinyl CoA synthetase enzyme, one molecule of ATP is produced, and this here uh, inorganic phosphate also take part in this reaction, and this coenzyme here removed and provide energy for the synthesis of ATP. In animal cell, it here in this step GTP is converted into GTP, but in plant here ADP is converted into ATP. So this is a substrate level phosphorylation. So one molecule of ATP is produced here in the form of here substrate level phosphorylation. Here then its regeneration phase is started here. Succinyl CoA is now being converted into succinate, which is a 4-carbon compound, and this 4-carbon compound is rearranged into a oxaloacetate here, that is again regenerate the oxaloacetate for acetyl CoA. So succinate will be converted into succinate dehydrogenase. That is a rearrangement. Here you can see that this CH2 here, one hydrogen will be removed and double bond produced here, and compound is fumarate, and one of its another coenzyme, that is FAD, that is reduced in the form of FADH2, that fumarate. Then this fumarate will be rearranged by adding water, converted into, in the presence of enzyme fumarase, converted into malate, and that malate again, that is a rearrangement and release of a hydrogen, which will be captured by NAD and formed NADH plus H. So oxaloacetate is now again generated for the next cycle of a for the next capturing the acetyl CoA. So here malate dehydrogenase is the enzyme. So if we look into a whole process of Krebs cycle, we will find here that we will obtain NADH here, second NADH here, third NADH here. These NADH are high energy molecules which will deliver to the electron transport chain, and from where energy in the form of ATP will be produced. And one FAD also reduced here, and one molecule of ATP is also produced in the form of substrate level phosphorylation. So as one molecule of acetyl CoA will enter, it will produce 3 NADH, 1 FAD, FADH2 and 1 ATP molecule. Keep in mind, if one molecule of a glucose will produce 2 molecules of pyruvic acid and 2 molecules of acetyl CoA. So with the 1 molecule of glucose, as it will produce 2 acetyl CoA, so this cycle will operate in a 2 time. So all the products will be multiplied by 2. So we will discuss its energetics in the next slide. So here is the energetics of the Krebs cycle. Here oxidation of these 3 NADH in one cycle of acetyl CoA, 3 NADH will be produced, and when these 3 NADH move into the electron transport chain, it will produce a 9 ATP molecule in the form of oxidative phosphorylation, as 3 ATP molecule will be produced by the oxidation of a 1 NADH. So 9 ATP molecule will be produced in this process, and keep in mind 1 FAD in one cycle is produced, that will produce 2 ATP molecule, and 1 ATP molecule in the form of substrate level phosphorylation is produced. So total 12 molecules are produced in 1 acetyl CoA, and keep in mind if we have a one glucose molecule will produce 2 acetyl CoA, so total count will be 24 molecule, and this will be discussed at the end. We will all again discuss the energetics of the whole aerobic respiration. That's all about today. In the next lecture we will discuss the oxidative phosphorylation and electron transport chain. Thank you very much.